Hey guys, welcome back. We're going to go ahead and keep on progressing through aerobic respiration. We left off last time at the end of glycolysis. Hopefully you recall our products of glycolysis, which we left off with 2 pyruvates. We had 2 ATP that we netted, and we had 2 NADH, which we're going to use in just a little bit. So I want to kind of start this one off with that knowledge. Quick review. What were the inputs of glycolysis? Hopefully you recall that would be glucose and 2 ATP to get the thing started. And then where in the cell does glycolysis occur? It's another thing you should know. Hopefully you're thinking cytoplasm because that's where glycolysis occurs. So we finished glycolysis. It took 2 ATP to get started. We made 4 ATP at the end of it. So we netted 2. And we're left with this weird little 3-carbon molecule called pyruvate, and we're not really sure what to do with him. So let's progress on with aerobic respiration, and we will find out what we're going to do with him. So the second stage of aerobic respiration is we're going to complete the breakdown of glucose, because we've got him down to 3 carbons, but that's not enough yet. We're going to move into the mitochondria for this. And it includes 2 sets of reactions. The first is we form acetyl-CoA, or acetyl coenzyme A, and then we move into the Krebs cycle. And we're going to have to go through each of these twice in order to break down one of those glucose molecules. So here's what it looks like. We did glycolysis out here. We made ATP, we made pyruvate. This is the outside of the mitochondria. This is the inner membrane inside of the inner membrane of the mitochondria. That's in here. This is 2 membranes, an inner membrane, an outer membrane, and this here is the inner membrane and the outer membrane. So now you know where we are. In the cytoplasm, we did our glycolysis. Now we're going to go ahead and take those 2 pyruvates from the cytoplasm and send them all the way through to the inner compartment of our mitochondria. And when we do, we're going to get our pyruvates into the Krebs cycle by forming acetyl-CoA. And then when that's finished, we're going to end up making carbon dioxide for waste. Carbon dioxide is going to diffuse out of our mitochondria, out of our cytoplasm, into a capillary, into a red blood cell. It's going to be carried to our lungs. We're going to exhale it, and it's going to be gone as a waste product. And then plants can use that later, hopefully, is the idea. So we're going to get a whole bunch of reduced coenzymes. We're going to get NADH out of this, which is good. We're going to get FADH2 out of this, which is good. And they will bring those electrons and hydrogens into our third stage, which we'll use later. And we're going to make just a little bit of ATP. Not a lot, 2. Kind of like the last stage. We made 2. It's not a lot, but it's better than none. So let's take a look at how this process works. We're going to start with acetyl-CoA formation. So in the inner compartment of the mitochondrion, some enzymes are going to split that pyruvate. And when they split it, they're going to form acetyl-CoA, and they're going to form carbon dioxide. So there's one of our carbons. We were down to that 3-carbon molecule, and now we're down to a 2-carbon molecule because we lost CO2 as waste. And then we're going to form NADH when we form acetyl-CoA. So we get acetyl-CoA and NADH out of that pyruvate. So now we've got acetyl-CoA, which is good. And now we're going to take that acetyl-CoA straight into the Krebs cycle. And the Krebs cycle is a bunch of enzyme-mediated reactions that break down one acetyl-CoA into 2 of these carbon dioxides. And these are just going to be waste, but we're going to take this acetyl-CoA and we're going to make a lot of useful stuff out of that acetyl-CoA. The reason we call it a cycle is because we start with oxaloacetate and we end with oxaloacetate. And so our product is also one of our reactants. Oxaloacetate is very closely related to oxalic acid, the chemical that gives rhubarb its tart flavor. And for those of you who were in botany, also Oxalis stricta, that gray-green wood sorrel that is so delicious, has that oxalic acid in it as well. And so we see this in a few places. But we use that, we regenerate that, that's why it's a cycle. And then we're going to make NADH. We're going to make FADH2, 3 of these, 1 of these, and you'll recall from the last slide that we're supposed to end with 8 NADH, 2 FADH2. And now we're seeing where those are coming from because we have to make 2 turns of this cycle. So we have the NADH that we previously made. 2 turns of this would put us at 6. 2 turns of this would put us at 2. And so it puts us back at our original predicted NADH and FADH concentrations. And we're going to make 1 ATP. So then 2 turns of the cycle makes 2 ATP. So products of Krebs cycle: carbon dioxide, NADH, FADH2, ATP. Important stuff. And here's what it looks like. It's a mess. Man, it is a mess. Here we go. Pyruvate comes in, carbon dioxide leaves, we make NADH. That's the missing 2 to make our 8 total. Pyruvate, once we've cleaved this carbon off to make our CO2, we get our 2-carbon acetyl coenzyme A. Acetyl-CoA makes a citrate. Our citrate loses a carbon and makes NADH, good product for later. We lose another carbon, make another NADH, good product for later. We make ATP, we make FADH2, another good product for later. NADH, another good product for later. And when it's done, this process of swapping carbons and phosphates, we're back to oxaloacetate. And at that point, we've got another acetyl-CoA ready in the inside of the mitochondria, and we start it all over again. So you could say, what's our inputs and outputs is one way to look at it. Where's our location? Inner compartment of the mitochondria. What's our inputs? Acetyl-CoA, oxaloacetate. What's our output? NADH, FADH2, and ATP. That's one way to look at that. So that's the Krebs cycle. Here it's step by step. An enzyme splits that pyruvate into coenzyme A, into a 2-carbon acetyl group, and that CO2. The coenzyme A binds to that acetyl group, and we form acetyl-CoA. And then NAD plus grabs that released hydrogen and makes NADH with 2 electrons. So then our Krebs cycle starts, because this was just our acetyl-CoA formation. That was step 1. Krebs cycle step 2 definitely gets a bit more attention. The Krebs cycle starts as one of the carbons from acetyl-CoA is transferred to oxaloacetate, and we get a citrate, and the coenzyme A is regenerated for the next turn. 3 carbon atoms removed, leaves a cell of CO2. NAD combines with the released hydrogen, and we make NADH. Then another carbon atom is removed, and another NADH forms with that hydrogen that's released when that happens. And now pyruvate's 3 carbon atoms have exited the cell as CO2. There's 1 CO2, CO2 number 2, CO2 number 3, and they're all out of there. So step 5, 1 ATP forms because we phosphorylate him. Step 6, the coenzyme FAD combines with some hydrogen ions and becomes FADH2. Step 7, NAD plus does the same thing and forms NADH. Step 8, we are back to oxaloacetate. We need more carbons to make this run of handing off hydrogens again, and we're going to get those carbons from here. So we add those carbons in, and we make this whole circle again of handing off hydrogens by making that carbon dioxide. So that waste product that we just think of as waste is actually really important, because when he breaks off of here, that's what leaves the spare hydrogen to make NADH and FADH2. And those are really important characters. So that's the Krebs cycle. Now, here they are together. A similar cartoon for the same process. They're just showing this one, they added the flow of electrons in with the hydrogens, but really the process is the same. So we're going to stop there. The next video we do will be our 3rd stage of aerobic respiration. So thanks for your time.